Sushruta for NEET is now available on the Google Play Store. Try it out for free.
Good morning, everybody. Hearty welcome to JnanaMarga URA class. Let us continue with the biotechnology. I have started just taking up the steps followed in the recombinant DNA technology. I have mentioned about origin of replication. Origin of replication is a sequence of nitrogen base from where replication starts. And any piece of DNA, when linked to this sequence, can be made to replicate within the host cell. So foreign DNA, alien DNA has to be linked to this ORI. This sequence is also responsible for controlling the copy number of the linked DNA. How many number of that linked DNA should be produced? The sequence is also responsible for controlling the copy number. How many copies of that is required? That also is concerned with the origin of replication. So if one wants to recover many copies of the target DNA, it should be cloned in a vector whose origin support high copy number. Right? So now I have mentioned about the selectable marker. What is the selectable marker? Selectable marker is the region where our gene of interest is ligated. So in addition to ORI, where origin of replication starts, the vector requires a selectable marker, a G area, a marker, a reporter gene, which helps in identifying and eliminating non-transformants and selectively permitting the growth of transformants. You must know what is a transformant and what is a non-transformant. To any of the bacteria, if vector is introduced, that bacteria is called a transformant bacteria. Transformed bacteria. If it is not included, if that plasmid, if any foreign DNA is always introduced into the bacteria through plasmid. If this plasmid is not introduced into the bacteria, if the plasmid is not taken up by the bacteria, it is not transformed. So please remember transformed and non-transformed. What is a transformed bacterial cell here? Transformed bacterial cell is a bacteria which has plasmid with or without recombinant DNA, which has plasmid from other source, not of its own, which has plasmid of its own. So whenever we introduce the plasmid into a bacteria, that bacteria is called transformed bacterial cell. Without that plasmid, non-transformed bacterial cell. Transformation is a procedure through which a piece of DNA is introduced into a host bacterium. And how we are introducing? Through plasmid. Okay. Why we introduce it through plasmid? Why not directly introduce it to the host bacterium? Who will tell me? Why that we introduce bacterium through the plasmid? Why not directly put it into the host bacterium, into the bacterium, host bacterium? Come on, who will tell me? Who will answer this question? Hello, are you hearing me? Are you hearing me? You know the answer. Hello, you know the answer. Kavita, can you tell me? Or who is going to tell me?
Yes, sir.
Yeah, tell me, Kavita. Kavi,
Yes, sir.
tell me. Why that we are not directly adding this DNA into the host bacterium? Why we are adding it through plasmid? Who will tell me? Raise your hand. I hope you know the answer. Please don't keep quiet. Hello? Janani, can you tell me? Hello? Janani, Janani, I'm waiting for you. I can't hear you. I can't hear you. You don't know. Navya, this I have told in the very first class.
Sir, is
Can you
it
start?
because plasmid is having the property of replicating?
Pardon? DNA may replicate.
No, sir. Plasmid is having the property of autonomously replicating. So it replicates a large copy of a DNA.
Yeah, it replicates one thing. There are two things, Smita. One, if at all you add this DNA directly into host bacterium, it is only in that host bacterium only it will be there. It will not be passed to, it will not be passed to next generation. It will be there in that particular cell only. It will not go to the next generation. It will not multiply. So here, the plasmid is an autonomous body which has a self-replicating ability and it goes on increasing a high copy number. Yes, very good, Smita. Star of the day. Okay, star of the day. Now, that is why we wanted to see that this is incorporated to the plasmid. Or transformation is the passage of foreign DNA or plasmid containing the passenger DNA into host cell. Right? Now, normally the genes encoding resistance to antibiotics we have in some bacteria, not in all. In some bacteria, there will be some gene, and these genes always developed developed resistance to antibiotics. If you put these bacteria with a resistant gene in antibiotics like ampicillin, chloramphenicol, tetracycline, kanamycin, etc., nothing happens to them. Otherwise, if at all treat with antibiotics kill them, kill them. So we get antibiotics. We get antibiotics. So to kill these organisms, kill these bacteria. Okay. So normally the genes encoding resistance to antibiotics, that means ampicillin, chloramphenicol, tetracycline, clindamycin, etc., are considered useful selectable marker for E. coli. Selectable marker for E. coli. One thing that you remember, the normal E. coli cell do not carry any resistance against any of these antibiotics. No, it has no resistance gene at all. E. coli has plasmid, no doubt about it, but it has no any resistance gene to any of these chemicals, any of these antibiotics. Ampicillin, chloramphenicol, tetracycline, kanamycin. You'll be asked which of these antibiotics the some bacteria possess resistance. That means how they possess, how these genes do. The gene in this bacteria that produces some protein, and these proteins act as a resistance. They prevent they prevent these antibodies killing these bacteria. So they have resistance power. How these bacteria develop resistance power? Because they form some protein in their body, and that proteins resist against these antibiotics. They make these antibiotics not to work. No. So now these genes encoding resistant to antibiotics. Encoding means genes deciding or genes coding for relevant protein. You know, genes direct the nitrogen bases in the DNA, direct mRNA. mRNA goes to the cytoplasm, makes the relevant amino acids to come there. We have already studied the transcription and the translation. So that is what is encoding. The genes encoding means that the genes becoming responsible to produce a particular protein encoding resistant to antibiotics are considered as a useful selectable marker. And E. coli has no such a resistant gene at all. Now let us look into cloning site or recognition site or also known as restriction sites. What is this? In order to link the alien DNA in order to link the gene of interest, in order to link the foreign DNA, in order to link the alien DNA, the vector needs to have very few, more probably, preferably single recognition site. This recognition site for the commonly used restriction enzyme. It should have single recognition site for the commonly used restriction enzyme. Whatever commonly used restriction enzymes are there, on the other day I have mentioned about this, and these are the commonly used recognition sequences for these restriction enzymes. Okay, so you should have recognition site for the common user restriction enzymes. Now let us see. That is what the sequences have shown you. Recognition site. What is it? Presence of more than one recognition site? Recognition site, or cloning site, or and this. It is there inside the selectable marker. I'll make it very clear what are the differences between all these. Presence of more than one recognition site within the vector will generate several fragments which will complicate the gene cloning. Now see here. This is what you have. What is this called? What is this called? What is this called? So this is a PBR 322. It is a vector. It is a plasmid in E. coli cloning vector PBR 322 showing restriction sites. These are all the restriction sites. Hint 3, EcoR1, okay, BAM H1. These are all recognition site. And here, BAM H1 has antibiotic resistance gene. Antibiotic resistant gene, AMPR, ampicillin resistant gene, tetracycline resistant gene, AMPR, TETR. Okay, and at the same time, you come across another area, what is called ROP, ROP. ROP codes for proteins involved in the replication of the plasmid itself. If this plasmid itself has to replicate, this functions ROP. It enables the plasmid to duplicate whatever number it wants. So this, I repeat, this BAMH1 is the restriction site. Hind is another restriction site. SAL, that PVV1 is, and in this BAMH1 restriction site, you do have recognition sequence for restriction enzyme BAMH1. So BAMH1 is a restriction site also. BAMH1 here, it is a restriction enzyme also. I hope you understand my point. BAMH1 is a restriction site, or this is a restriction site, recognition site, cloning site. They are all one and the same. And in this BMH1, you can find what gene? You can find gene, gene which has a resistance to tetracycline. You can find the gene here where you have PBU1, PST1. What are the PBU1 and PST1? These are, what are these? What are these? These are restriction site with the restriction enzyme with the recognition sequence for these. Right? Understood? Are you following me? Any doubt here? Tanusri?
No.
No? Namrata?
No, sir.
Nadira?
No, sir.
Jyoti?
Yes.
Siddesh? Harshavardhan?
Okay.
Charan?
Yes, sir.
Charan?
Yes, sir.
Okay. Now, let us look into this. Can you see here? What is this? This is a gene donor. One bacterium, let us consider from where we wanted to isolate the gene. And this is a plasmid donor. It has a donated plasmid. And this gene donor, from this gene donor, we got a piece of DNA which we are interested isolated. And this also got cut here. Where it is got cut, plasmid got cut open with the restriction enzyme, right? And this has to be joined here. So isolated gene or gene of interest, desired gene, alien gene is got ligated to this plasmid. What is the enzyme involved in that? Enzyme involved is a DNA ligase. After that, this plasmid is introduced into the bacteria. So what you call this bacteria now? Transformed bacteria. Transformed bacteria. So transformed bacteria is the bacteria to which this particular plasmid, a foreign plasmid, is introduced into the cell. Now this transformed bacteria is with the recombinant DNA, with the DNA piece ligated, foreign DNA ligated to that, genoprinted like that. So this is therefore, this bacterial cell is called transformed bacteria with the recombinant type. It is a transformed recombinant bacteria. I hope you understand. Once this plasmid is with the recombinant DNA is inserted, this now it replicates. It has a self-replication capacity and it produces whatever number it wants. That plasmid gets replicated, whatever number of DNA, mRNA are produced, and whatever protein that's required that gets produced. Okay, clear? Understood? So now we are doing the recombinant DNA technology. Why? We are doing it to get what you want. All these things are done to get what you want. Whatever you want, generally, that will be always in the form of a protein. It will be in the form of protein. Ultimately, we know gene expression is nothing but the production for element of protein. Everything is protein maya. So this recombinant DNA technology also enables us to get what you want in the form of protein. Understood? Clear? Clear? Now, the ligation of alien DNA is carried out at a restriction site. Ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistance gene. One of the two antibiotic resistance gene. Which are these two? Which are the two antibiotic resistance gene? Which are the two antibiotic resistance gene? Canisa? Canisa?
Yes, sir.
Tell me. Which are the two antibiotic resistance genes? Rafa?
Yes, sir.
Which are the two antibiotic resistant genes?
Ampicillin.
And
And TETR.
what is that ETR?
Tetracycline.
Tetracycline resistant gene. Canisa? Canisa?
Yes, sir.
Why you are keeping quiet?
I answered, sir.
Antibiotic resistant gene, that is a ampicillin resistant gene, tetracycline resistant gene. So ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant genes, and these two are always used. Ampicillin resistance gene site and tetracycline resistance gene site. So, for example, you can ligate a foreign DNA at the BAM-H1. What is that at BAM-H1? BAM-H1 site of tetracyclic resistant gene in the vector PBR322. What is the ampicillin resistant gene? Which is the site of ampicillin resistant gene? Which is the site of ampicillin resistant gene? Tanusri? Tanusri? Which is the site?
1 and PST1.
Good. Okay. It is a PVU1 and PST1. Okay, right. It is clear now. So the recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA. Very important. Very important. Whenever you have this resistant gene, if something is added in that region, if something is ligated to that region, that gene loses its capacity of resistance. Remember, very important term, very important sentence. Whenever we just ligate foreign DNA, alien DNA in the antibiotic resistant gene site, that particular antigen resistant gene it loses its ability of resistance. So the recombinant plasmid will lose tetracycline resistance. Recombinant plasmid. What do you mean by recombinant plasmid? A recombinant plasmid is a plasmid with the alien piece of DNA. The recombinant plasmids will lose tetracycline resistance due to insertion of a foreign DNA, but can still be selected out, from non-recombinant ones by plating the transformants on a tetracycline containing medium. How? The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinants. Can be selected out from non-recombinants one by plating the transformants on a tetracycline containing medium. What happens? If you put it in a tetracycline containing medium, what will happen to this? What will happen to this? Hello? Are you understanding my point? Now, please listen to me. I want you to respond, otherwise I cannot make out whether you understood or not. The recombinant plasmid, what do you mean by recombinant plasmid? A recombinant plasmid is the one where the gene of interest is ligated to the site of antibiotic resistance gene. The recombinant plasmids will lose tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on a tetracycline containing medium. That is, the recombinant plasmid does not possess tetracycline resistance, but continues to have amphigenin resistance. Are you understanding this point? Please read this and try to understand. Now, we are putting this recombinant plasmids, in two mediums, in one ampicillin, in another tetracycline. Okay? We have recombinant plasmids. Where it has come? This recombinant plasmids got incorporated into bacteria. Now I am putting this bacteria in some of the bacteria in the ampicillin medium, in some of the bacteria in the tetracycline medium. And I wanted to find out which are the transformed and which are the non-transformed. How can I decide? Which are the recombinant type, which are the non-recombinant type? How I can decide? Please don't keep quiet. Now we have introduced this plasmid, recombinant plasmid, into bacteria, into some bacteria. Okay, some population bacteria, we have incorporated this recombinant plasmid into this bacteria. That means whatever the plasmid, it got alien piece of DNA, it got introduced into the bacteria. Now I wanted to find out, okay, which of these bacteria are with the recombinant one, which are with the non-recombinant one. It may not be that this plasmid, you have introduced into the plasmid. This plasmid with the DNA is introduced into the bacteria. Bacteria has its own plasmid also. It is not that bacteria is without any plasmid. One of the most important feature of the bacterium is possession of plasmid. Can you make out which are the one with the recombinant, which are the one with the non-recombinant? Hello, please try to understand and answer my question. Who will answer my question? I repeat again, the recombinant plasmids will lose the tetracycline resistance due to insertion of foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on tetracycline containing medium. That is, the recombinant plasmid does not possess the tetracycline resistance, but continues to have amphigenin resistance. What do you mean by this? How you answer my question? How you decided this is with the recombinant DNA It is without recombinant, not recombinant one. Bacteria, recombinant one, not recombinant one. Jyoti,
Yes, sir.
can you answer this question? Or you want me to explain this again?
Sir,
Hello?
sir,
Hello?
sir, the recombinant DNA will not show resistance against tetracycline, but it will show resistance against ampicillin.
That means how you say? How you say that it shows resistance means how you say? So now you are putting bacteria. You are putting bacteria in two media. What is there in one media? Antibiotic. Hello.
Ampicillin.
Huh? Yeah. Yeah. And another medium? Tetracycline.
Tetracycline.
Now you tell me whether this bacteria will survive in both. Hello?
No, sir.
Now you have bacteria, some bacteria with the recombinant DNA, some bacteria without the recombinant DNA. What do you mean by that? Some bacteria the recombinant plasmid, some bacteria without recombinant plasmid. Okay. So, which one will survive in ampicillin medium? Whether all bacterium, with all bacterium survive in ampicillin medium?
No.
Why?
Recombinant DNA will not survive.
My question is, you have added this recombinant DNA where? You have added this alien DNA where? In which site? Hello? Now, this gene of interest is ligated to which place? There are two ampicillin resistance, antibiotic resistance gene. One is ampicillin resistance gene. Another one? Yeah.
Tetracycline.
Yeah.
Tetracycline.
Now, you have added this piece of DNA to which area?
Tetracycline resistant.
Tetracycline.
Tetracycline.
Yeah. So that what has happened? That particular plasmid with the gene of interest ligated to tetracycline loses the power of resistance. Okay. Now you are putting you are putting bacteria in ampicillin medium. Do all bacteria survive or do all bacteria die?
All survive in ampicillin.
All survive in ampicillin. Can you say who said it like that? All survive in ampicillin.
Not
And in
all.
tetracycline medium,
The recombinant one will not survive.
the recombinant will not survive, but non-recombinant will
Survive.
survive. So can we now can you understand then? The recombinant plasmids will lose the tetracycline resistance due to insertion of foreign DNA. Due to insertion of foreign DNA, recombinant plasmid will lose the tetracycline resistance due to insertion of foreign DNA. So what will happen? These plasmids with the recombinant DNA or these bacteria with the recombinant DNA, they are going to die, but can still be selected out from non-recombinant ones. What is that non-recombinant ones? Without DNA ligated to their antibiotic resistant gene. Once by plating the transformants on tetracycline containing medium. If at all we put a transformant on the tetracycline containing medium, what will happen? Whether they will survive or die. Hello? If the transformants are put in a tetracycline containing medium, do they survive or do they die? Why you are keeping quiet? Sarvattam, see, you must respond. Sir, we have not understood. Or we know the answer, this answer. Otherwise, how I can know? How many of you have not understood? Raise your hand.
Sir, the ending transformant part.
Pardon, Raveshiti?
Sir, the ending transformant part I didn't understand.
See, I shall repeat again. Please listen to me. All of you, hands down. Please listen to me. Please listen to me. Are you ready? Now, where I have to add this gene of interest? I want to add a gene of interest into a plasmid. To which part I have to add?
Ampicillin and tetracycline.
See the sure right. We have selectable marker. Which are the selectable marker? One selectable marker is ampicillin resistant gene. Another selectable marker is tetracycline resistant gene. Is it clear? Is it clear?
Yes, sir.
So
Yes, sir.
I have no other area. I have to put my gene of interest, the gene of interest, whatever I am interested. I have to put it in the plasmid. Where I have to put? I have to put in the selectable marker gene. I will tell you why I have to put it there later. I have to put in these two areas, either ampicillin resistant gene area or tetracycline resistant gene area. Understood? This is the point. I have no other place to introduce the gene. This is the recognition site, restriction site. Where I have to ligate? One. After ligating, after putting in that particular area, tying up in that particular area, that gene will lose the power of resistance. That gene will lose the power of resistance. If at all I ligate this to ampicillin resistant gene, that gene will lose the power of resistance. That means if you put that bacteria in ampicillin medium, ampicillin medium is a culture medium where ampicillin is antibiotic is there. We are just putting that bacteria in that medium. What will happen to that bacteria?
Survival.
Survives or dies?
It dies.
Survive.
Savitri?
Yes, sir.
It survives or dies?
Survives.
Why? I'm repeating it. I'm repeating it. We have ampicillin resistant gene one part, tetracycline resistant gene another part. These are the two in a circle, in a cycle like this, in a circular plasmid. You have one part ampicillin resistant gene, another part tetracycline resistant gene. Let us see that. If I want to add a piece of DNA, I have to put DNA either here or here only, nowhere else. And it is near to eco-RI. I have to either put here or I have to put it here. The moment I introduce this foreign DNA in ampicillin resistant gene area, ampicillin resistance power is lost. If I ligate in a tetracycline resistant gene area, tetracycline resistant power is lost. The bacteria which has the power to resist this tetracycline antibiotic is lost. Now what I have asked, I have put, now when it is lost means, if at all you put that bacteria in that medium, the bacteria gets killed. If at all when the ampicillin resistance power is lost, if at all you put that bacteria in ampicillin medium, the bacteria will be killed because it has no resistance power. It will die. In the same way it is for the tetracycline. Now what I am telling, I have taken a DNA, I have put that DNA into the ampicillin resistant gene area site, and later I am putting it in a medium containing ampicillin. what happens to that bacteria? Whether that bacteria survives or die?
It dies.
It dies.
Savitri,
Dice, sir.
understood?
Yes, sir.
Yeah. Now what we have done? We have added this gene of interest. We have added this alien piece to tetracycline area. Tetracycline having area. Tetracycline resistant in area. After that, if at all, now we want to distinguish recombinant type, non-recombinant type. So you have two types of plasmid, recombinant, non-recombinant. Which one will survive? Which one will survive? Recombinant, non-recombinant in amphigyline? Which one will survive in a tetracycline?
Recombinants.
Vismita?
Recombinants will survive in ampicillin.
Recombinants will survive in amphigyline and die in
Tetracycline.
tetracycline. Non-recombinants will survive.
In tetracycline
Yeah.
and die in ampicillin.
Why? Now 1 minute. Is it correct? Rafa? Rafa? Aisha? Blessy? I repeat again. Vismita said, recombinants will die in
Tetracycline.
tetracycline, survive in
Ampicillin.
ampicillin. So by this you can distinguish which are the recombinant, which are the non-recombinant. Am I right? Am I right?
Yes, sir.
You can distinguish which is the recombinant, which is the non-recombinant. My point is that now you have non-recombinant plasmids. Non-recombinant plasmids, are they going to die or are they going to survive when you put them in ampicillin medium?
Survive.
Survive.
Vismita,
Yes, sir.
have you followed? No? Huh?
Yes, sir.
Have you followed?
Yes, sir. Yes, sir.
You are upset.
No, sir.
So, recombinants will survive. Who will tell me why it will survive? Harshvardhan? Sarottam? Siddesh?
Yes, sir.
Why they survive?
Because it has both resistant genes.
They got both resistance gene functional. Am I right? Am I right?
Yes, sir.
Because both are functional, it has a resistance for ampicillin, it has a resistance for tetracycline. So non-recombinants are going to survive in both medium. Have you understood now? Duti, Rakesh,
Yes, sir.
Sakshi, Namrata, have you again I'm going back to the sentence. Look here. The recombinant plasmids will lose tetracycline resistance due to insertion foreign DNA, but can still be selected out from non-recombinant ones by plating the transformants on tetracycline containing medium. If you place the transformant, if you put the transformants on tetracycline containing medium, what will happen to the transformants? Will it die or survive?
Die.
Die. It will die. That is, the recombinant plasmid does not possess the tetracycline resistance, but continues to have ampicillin resistance. So, the recombinant plasmid does not possess tetracycline resistance. You can add, so they die, but continue to have ampicillin resistance. But in ampicillin medium, they are going to survive. So you are putting them in the ampicillin and in tetracycline. Whatever they are surviving in ampicillin, you are selecting because they possess They possess recombinant DNA. Understood? Hello? Hello, please raise your hand if at all you understood. Otherwise, I'll stick on to that and I'll do this one only. Because I have to. Yes, please raise your hand. Hold your hand. Because I'm seeing here, I'm still not seeing Aisha's. Okay. Okay, that 1 minute. It will not show me how many people have raised their hand. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. Okay, right. So I shall go ahead. Okay, now, right? If it is not understood, you can call me. I will explain that again wherever it is possible. Okay. Now, the transformants growing in on amphibian containing medium are then transferred on a medium containing tetracycline. The transformants growing on amphibian containing medium are then transferred to a medium containing tetracycline. What will happen to that? If they are transformed to tetracycline medium, what will happen? Kenisa?
Die.
They die. Okay, right. Now, the recombinants will grow in amphibian containing medium, but not on that containing tetracycline. But the non-recombinants will grow on the medium containing both antibiotics, ampicillin and tetracycline. I hope it is clear now to you. In this case, one antibiotic resistant gene helps in selecting transformant, whereas the other antibiotic resistant gene gets inactivated due to insertion of alien DNA that helps in selection of recombinant. So whichever the another one inactivated due to insertion of alien DNA that helps in selection of recombinants. Recombinants. Now you have the medium. If in the antibiotic resist in the ampicillin antibiotic, if it is they survive, that means they got that recombinant DNA in the tetracycline resistant gene area. Okay, so this is how it is selected. Look here, you just now I want you to look into that, and I want you to explain. I am not going to explain. I will explain later. You look into this and come forward. I will give you a 5-star. I will give you the award 5-star. You have to explain. If you raise your hand, I can't see because I am in the full screen. You have to call me and then you have to explain.
Sir,
Yes, who is this?
Navya.
Yeah, tell me now. Yeah.
So the ligation takes place in the tetracycline resistant site.
Hmm.
So it has lost the ability uh, it has lost the ability of resistance against tetracycline. That is why the recombinant is not tetracycline resistant. So when put in that medium, it might die.
Okay, very good, very good. So, Navya, I am explaining in another way. See, all of you tell me, what is this? What is this?
Plasmid.
A plasmid. And this, what is this plasmid called? PBR
PBR 322.
322. And it has ampicillin resistant gene and also tetracycline resistant gene. Ampicillin resistant gene, tetracycline resistant gene. So what is done here now to this particular area, what is done now? We have, what is this? Gene of interest. This gene of interest, or desired gene is ligated here. It is attached here. See, one piece of this blue here, another piece of blue here, and it is ligated here. So what do we call this now? What do you call this plasmid? Recombinant. Why it is recombinant? Because gene of interest is added. Recombinant DNA. Right? Now, this, what is the tetracycline resistance? This plasmid, whether it has a resistance to ampicillin, whether it has still a resistance to ampicillin, yes or no?
Yes, sir.
Yes. Ampicillin resistant, yes. Whether does it have tetracycline resistance?
No.
No, no, it has no tetracycline resistance. Okay, no. All right. Now, what do you do? Now, whatever gene of interest is added, it is removed. It is removed, religated, religated, ligated one, religated means we have removed and we have joined it again. So now what happens? Does it show ampicillin resistance?
Yes, sir.
Yes. Does it show tetracycline resistance?
Yes.
Yes. So you have understood this clearly? Okay. ಅರ್ಥ ಆಯ್ತಮ್ಮ ತನುಶ್ರೀ have followed
Yes, sir.
Jayashree,
Yes, sir.
Nadira,
Yes, sir.
Siddesh,
Yes, sir.
Rakesh,
Yes, sir.
Lohit,
Yes, sir.
Prithvi,
Yes, sir.
Kanisa, Kanisa, Kanisa.
Yes, sir.
Why I am asking so many boys and girls understood? Why I am calling these all you all and trying to find out whether you understood or not? Rafa, Aisha, Namrata,
Yes, sir.
why I am calling by name and asking you all?
If we are listening or we are active, sir.
One thing, whether you are listening or active or whether you are there or not. Okay? For both the reason. For both the reason. Okay? Do you like this asking like this? Rafa?
Yes, sir.
Yes? Charan?
Yes, sir.
Yes, okay. Now, see this. I think this I don't want. Let us leave this one. So, this you understand. Insertional inactivation. What do you mean by insertion inactivation? Always ask a question. Insertion inactivation is the inactivation of a gene upon insertion of another gene inside in its place or within its coding sequence. Whenever we just insert a gene in the coding sequence of another gene, that gene becomes inactive. Whatever the gene that got inserted, that gene becomes inactive. This helps in the selection of recombinant colonies in recombinant DNA technology. Okay, right. Now, selection of recombinants due to inactivation of antibiotics is a cumbersome procedure. We have to put it in that plate, this plate. Again, we have to find out selection of recombinants due to inactivation of antibiotics. Say we have said tetracycline resistant gene became inactive due to inactivation of antibiotics is a cumbersome procedure because it requires a simultaneous plating on two plates having different antibiotics. You have to put once in ampicillin, another you have to put it in a tetracycline, and you have to find in which it will die, in which it will survive. It is a cumbersome process. To solve this problem, therefore, alternative selectable markers have been developed, which differentiate recombinants from non-recombinants on the basis of their ability to produce a color in the presence of a chromogenic substrate. I repeat again. So, instead of going for putting it in two different plates and then selecting, it is a time-consuming cumbersome. Another alternative celebrable marker have been developed which differentiates the recombinants from the non-recombinants on the basis of color, on the basis of ability to produce color in the presence of a color substance, chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta galactosidase. In this, a recombinant DNA is inserted within the coding sequence of an enzyme, beta galactosidase. This results into inactivation of the enzyme, which is referred to as insertional inactivation. I repeat, okay, I'll come to this point. I'll go forward and come back. Okay, now, look here. Look here. This is a plasmid. Okay, and this plasmid has already recombinant DNA. It is a recombinant plasmid. Recombinant plasmid, right? Now we wanted to find out this plasmid with the recombinant is called recombinant. Recombinant with the gene of interest inserted into that. Now we wanted to find out now how to distinguish a recombinant from non-recombinant. What do you mean by non-recombinant? A non-recombinant plasmid is the one where gene of interest is not added. It is not added. We wanted to distinguish a recombinant from non-recombinant. For that, what is done? We are adding another foreign DNA. To whom? To already recombinant plasmid, to a already plasmid with the recombinant DNA. We are adding one more foreign DNA. Where we are adding? We are adding in a place where we are adding in a place having lac Z gene. We are adding this foreign DNA into lac Z gene. Why lac Z gene? Why lac Z gene? Lac Z gene is considered what? Have you heard about lac Z gene? Anyone? Have you studied lac Z gene? It is concerned with the coding for. Can you remember Vismita? Have you heard about that? Have you heard about this gene before? Anyone? Anyone? Have you heard about this gene before? Hello? Have you heard about the gene lac Z before?
Yes. Structural
Who
gene.
is that? Who is that?
Rakesh.
No, Rakesh. In what context you heard?
Molecular biology.
No, lac operon. What is the function of lac Z?
It codes for beta galactosidase.
Who said it?
Vismita.
Vismita. Good. It codes for beta galactosidase. Remember, this beta galactosidase is required to make that chromogenic substance blue in color. Blue in color. Beta galactosidase enzyme is required to make these chromogenic substance into blue color. Now, what has happened? We have incorporated a foreign DNA in a LAC-Z gene. So, LAC-Z gene got disrupted. LAC-Z gene got disrupted. When a LAC-Z gene got disrupted, what will not be produced? What will not be produced? What will not be produced?
Beta galaxy.
Beta
galactosidase.
galactoside is not produced. When beta galactoside is not produced, the chromogenic substance will not be converted into
Blue.
blue color. See, now we have here A transformation. Foreign DNA enters the host cell. Now we have added this foreign DNA into LAC-Z, and this plasmid is entering into the host cell, bacteria. So it has come inside. Selecting cells that have received a plasmid. Selecting cells that have received a plasmid. So some of the cells receive this plasmid with the foreign DNA. Now, distinguishing cells carrying insert containing recombinant molecules from cells carrying vectors without insert. So you have some plasmid with the insert, some plasmid without the insert. So bacteria which gets a plasmid with insert will be blue in color, or bacteria which gets no insert will be blue in color. This is intact vector, no insert. LAC-Z gene is still functional. LAC-Z gene is intact. LAC-Z gene transcript. LAC-Z processes X galactosidase, and this X galactosidase it transforms blue pigment into chromogenic substance into blue pigment. And wherever this insert is there, LAC-Z gene is split by foreign DNA insert. No product of a beta LAC-Z gene, that is X galactosidase, so no blue color, so colorless. Understood? Again, I am explaining. The presence of a chromogenic substrate gives blue-colored colonies if the plasmid in bacteria does not have an insert. Presence of insert results into insertion inactivation of beta galactosidase, and colonies do not produce any color. Understood? Is it clear? Please tell me. You want me to explain again? You want to explain again?
Yes, sir.
Yeah, okay. Now, what we are doing, we wanted to differentiate recombinants from non-recombinants. What is a recombinant? Kanisa, what is recombinant?
Plasmid which has a desired gene.
Yeah, plasmid which have desired gene is recombinant. Plasmid without the recombinant gene is not without gene of interest is not recombinant. We just to find out what is done, a new technique is involved. In this, a recombinant DNA is inserted. A recombinant DNA, a piece of DNA is inserted within a coding sequence of an enzyme beta galactosidase. You know, you heard about LAC-Z. I repeat again, LAC-Z. This LAC-Z has this LAC-Z gene area. Here we have added one foreign DNA. The LAC-Z is very much required to just convert the chromogenic substance, just to convert to the chromogenic substrate into color. Chromogenic substance into color. When you put this in the chromogenic substrate, if the LAC-Z is intact, LAC-Z produces a beta galactosidase. This beta galactosidase converts this chromogenic substrate into color. Which color? Blue color. If the LAC-Z is not functional, the blue color will not be produced. The chromogenic substrate will be colorless. It will be colorless. So what we have done, this we have taken plasmid. What plasmid it is? What plasmid it is? This plasmid is already with the
Recombinant.
recombinant DNA. It is already a plasmid of recombinant type. To this plasmid, we wanted to know whether it's a plasmid is recombinant or non-recombinant. So you may have some plasmid with the recombinant. Let us have another plasmid here, side by side, without the recombinant DNA. It has no recombinant DNA. Now what we do, in this, both the recombinant and non-recombinant plasmid, we are going to add a gene, we are going to add a recombinant, a small piece of foreign gene into LAC-Z gene. The moment, why that LAC-Z gene is important? LAC-Z gene is concerned with the production of a beta-galactosidase enzyme. Beta-galactosidase is responsible for the formation of color. When you put this plasmid in a colored substrate, in a chromogenic color-producing substrate. Color-producing substrate will become colored if beta-galactosidase enzyme is produced by LAC-Z gene. So now what we have done? We have added the gene, we have ligated the gene, another gene, foreign DNA in LAC-Z gene. When the foreign DNA is introduced into the LAC-Z gene, later, we are putting recombinant DNA with the LAC-Z gene, and we have non-recombinant DNA with the LAC-Z gene disrupted. We are putting both these in chromogenic substance. Now you tell me, which particular plasmid will get blue color, which particular plasmid will not be blue color? Plasmid is the recombinant DNA.
Will get blue color.
Recombinant DNA having LAC-Z gene, it will be blue color. Without recombinant gene, without LAC-Z gene or LAC-Z gene disrupted, hello?
No blue color.
No color.
No. Look here, intact vector. What do you mean intact vector? No insert. Intact vector, no insert. Vector with insert. With insert. This is no insert means which one? No insert. What is that insert here?
Foreign DNA.
Pardon? Hello? Are you not understanding my point? Hello? Hello?
Yes, sir.
Viswita,
Yes, sir.
what is the doubt you have? Coming back again. Yeah, once again. I'll explain it once again. Now what we have? Selection of recombinants due to inactivation antibodies is difficult, so we are going for another method. Which one? We are now wanted to find out plasmid with the recombinant DNA, plasmid without the recombinant DNA. Plasmid with the recombinant DNA, plasmid without the recombinant DNA. For that, what they have done? They wanted to know whether it's a recombinant DNA, plasmid is with the recombinant DNA or plasmid is without the recombinant DNA. And we wanted to make sure that the plasmid is with the recombinant DNA. In order to find out plasmid with the recombinant DNA, they are going to do the experiment base. Therefore, alternative selectable markers have been developed which differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. So, in this, a recombinant DNA is inserted within the coding sequence of an enzyme. Recombinant DNA that already we have a plasmid with the gene of interest, whatever we have added to that. Already we have added to that. And so, here, in this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. This results into inactivation of the enzyme, which is referred to as insertion inactivation. So here, of course, this figure is not given in your NCERT book. Straight away going here. The presence of chromogenic substrate gives blue-colored colonies if the plasmid in bacteria does not have an insert. Blue colored colonies if the plasmid bacteria does not have an insert. Presence of an insert results in insertion inactivation of beta calypsidase and colonies do not produce any color. These are identified as recombinant colonies. Okay, I shall see another diagram. Can you see this? This I have showed earlier, and again I am showing you another. Okay, this may make you to understand better. Shall we go by step? Look here. The foreign DNA and the plasmid are cut. Plasmid, this is the plasmid, are cut with the same restriction enzyme, which recognizes a particular sequence of DNA called a restriction site. The restriction site occurs only once in the plasmid and is located within a lax gene and a gene necessary for metabolizing lactose. Okay, you added that foreign DNA into plasmid. The restriction enzyme creates a sticky end, right? This foreign DNA got added here. This foreign DNA got ligated here. The ligated cloning vector is transformed into a bacterial host strain that is amphigen sensitive and is missing lac Z gene from its genome. It is missing the lac Z gene from its genome because we have added one more into the lac Z area. Bacteria are grown on a medium containing ampicillin and X-gal. Ampicillin is also there and X-galactosidase is there. It is grown on media containing ampicillin and X-galactosidase, a chemical that is metabolized by the same pathway as lactose. The ampicillin kills bacteria without plasmid. Ampicillin kills bacteria without plasmid. Plasmids lacking the foreign insert have an intact Z gene and are able to metabolize X-galactosidase, releasing a dye that turns a colony blue. Plasmid with an insert have a disrupted X gene and produce white colonies. So here you have two types of colonies, one blue and one white. Blue colonies have plasmid without insert and white colonies have plasmids with the foreign insert. So now I think you are doubting, you wanted to know in what way it is going to decide about recombinant and non-recombinant. Hello? Hello?
By color.
Pardon?
Color.
ಏನದು ಹೇಳಿ I do not understand. I want to know what is your problem in understanding this. Which one color you are telling? Yes?
If we get blue color, it is recombinant.
If you get blue color, what is the indication?
Recombinant.
It is a recombinant. Recombinant. If no blue color?
Non-recombinant.
Is it?
Sir, if it is blue in color, it is non-recombinant, and if it is white in color, it is recombinant.
Yeah. So here see, this is vector. Vector. So already you know what is that? Vector is a plasmid. Okay, it is a recombinant plasmid. This recombinant plasmid with the recom gene is tested now. It is tested by using another recombinant gene in lac Z area. We have now, you have two types of plasmid. One recombinant plasmid with the recombinant gene. Another plasmid without recombinant gene. I have told you, whenever we are adding the plasmid to a bacterium will be having its own plasmid. Apart from this foreign plasmid, it will be having its own plasmid. So, you have two types of plasmid. One is its own, another one is Another one is a vector. A vector. Now you have to distinguish the vector and the own plasmid. For this, we are doing this. So now this is a recombinant plasmid. To this, what we have done? We have added a new DNA piece to the laxed. When new DNA piece is added to the laxed, now this laxed function is disrupted. As a result of which, the plasmid which is added to that, it will not be blue in color. The cell will not be blue in color, and it will be colorless. So we have added a piece of DNA to the laxed area, making the laxed non-functional. Coming back here, distinguishing cells carrying insert containing recombinant molecule from cells carrying vector without insert. Carrying vector without insert. With insert, without insert. Distinguishing cells carrying insert containing recombinant molecule, from cells carrying vector without insert. So here laxed is inserted. Here laxed is not inserted. Wherever the laxed inserted, laxed not inserted. Wherever the laxed is inserted, gene in the laxed is inserted, you will not see the color. Wherever it is not inserted, you will see the color. Intact vector, no insert. Laxed gene, it works, it transcripts, it makes this blue. Where there is no, it cannot make it, it cannot convert the chromatin substance into blue, so it will be white. If at all it is not understood, I shall give the time here. You please make it a point to read, try to understand. If you still have the doubt, we shall discuss this again in the afternoon session. Okay, the presence of chromogen substrate gives blue colored colonies if the plasmid bacteria does not have an insert. If the plasmid in the bacteria does not have the insert, presence of insert results in insertion inactivation. Whenever we add, it is a insertion inactivation. Insertion inactivation makes the gene not to function. Right? So, I'll be taking up this again in the afternoon if you want. Vectors for cloning genes in plants and animals. Okay, I shall stop this here. I will continue from here because let me make that particular point very clear to you. And I'll give some time for you to study. And let me take up in another way if you don't understand that. Now, I wanted to know how many of you understood this. If it is not understood, let me try in another way explaining that. No problem at all. Or can you tell me, if it is not understood, what part you have not understood? ಏನು ಅರ್ಥ ಆಗಿಲ್ಲ ಅದನ್ನು ಹೇಳಿ. ಎಷ್ಟು ಮಂದಿ ಅರ್ಥ ಆಗಿದೆ ಕೈ ಎತ್ತಿ. ಫಸ್ಟ್ ಯು ಟೆಲ್ ಮಿ ಸರ್, ವಿ ಅಂಡರ್ಸ್ಟುಡ್ ದಿಸ್. Hello? I will ask. Aisha? Blessy? Hello? You are not replying to me. Visvita?
Yes, sir.
Have you understood or not?
Sir, I understood, but that last part little bit confusing, sir.
Which part? Are you not keeping well?
No, sir, I'm having cold and cough.
Okay. Now let us talk later. Okay. Kanisa,
Yes, sir.
have you understood or not?
Little I understood, sir.
What confusion is there? You let me know about that so that I can understand. I can make some other way of explaining this.
That blue and white color I did not understand.
No, blue and light color. I shall make it very clear. Now what we want? We wanted to test one particular plasmid. What type of plasmid it is? Plasmid with the recombinant DNA. Okay, recombinant DNA. It is with the recombinant DNA. We wanted to find out whether this plasmid is with the recombinant DNA. For that, what we wanted to ascertain that it is recombinant DNA. In that plasmid, we are using another technique just to find out what we are doing. We are just interrupting lac Z area. There is one more part in a plasmid, lac Z. This lac Z is responsible for the enzyme production called beta galactosidase. The enzyme beta galactosidase is required. Beta galactosidase is required to convert a chromogenic substance into color blue. I repeat again. When we put this plasmid in a substrate containing chromogenic substance, it will get converted to blue because the lac Z area, normal lac Z area, it produces beta galactosidase. Beta galactosidase is required to convert a chromogenic substance into blue. Now what is done? We are disrupting lac Z area. What is done? In a lac Z area, a small piece of DNA got inserted, foreign DNA. When this DNA got inserted, lac Z becomes insertional inactivated. It becomes inactivated, insertion inactivated. What do you mean by insertion inactivation? By adding a piece of DNA to a gene, that the gene becomes inactive. So when it becomes insertion inactive, what happens now? Beta galactosidase is not produced. Beta galactosidase is not produced means chromogenic substance is not converted into color. Understood? Is it clear?
Yes, sir.
Yeah. So this enables us to know whether this is a recombinant type or non-recombinant type. So recombinant type or not recombinant type, we are testing with what? We are testing for lac Z. Lac Z shows this is a recombinant, this is a non-recombinant. Right? Understood? If the lac Z shows it is a recombinant, it is recombinant. This another gene, already we have inserted a gene, in ampicillin resistant area. That is also recombinant. Both are recombinant because we are testing with the recombinant gene. Right? I shall stop it here. We shall continue in the afternoon class. If you don't want, I will start from here itself. Okay? Right? See you.